And we're recording. Let's go review muscle PowerPoints. Wrong place. Here, I want to review some muscular processes and make sure they're clear. So we went through all of this. Okay, there's a WebAssign question. One muscle is surrounded by epimysium. That means upon the muscle. The skeletal muscle organ is connected to a tendon. It's a muscle is a bundle of fascicles. A fascicle is a bundle of muscle cells. So the fascicles are surrounded by perimysium, and the muscle fibers are surrounded by endomysium. These long filaments, that's what muscles are, filaments of fibers that can contract. That's all muscles do. They don't push, they pull. Okay, so I want to review that. And let's look here. So the entire muscle cell is surrounded by the endomysium. Inside is the sarcolemma. So this is single muscle fiber, means single muscle cell. So each muscle filament is made up of bundles of myofibrils, which are made up of thick and thin filaments. All of the muscle myofibrils are surrounded by sarcoplasmic reticulum and T tubules. And if you add the sarcoplasmic reticulum on either side of the T tubule, that's called the terminal cisterna. And the three of them, each part of the sarcoplasmic reticulum on either side of the T tubule is a terminal cisterna. Two terminal cisterna with the T tubule is called the triad. So the muscle has extensive connection to the exterior of the muscle fiber or muscle cell, and that's how sodium gets in and causes the release of calcium. So each muscle fiber is made up of repeating units of actin and myosin. The thick filaments are myosin, the thin filaments are actin. Notice how all of the thick filaments in blue are surrounded by the actin. So when a muscle contracts, these myosin heads are going to be energized. Calcium is going to come in, and it's going to bind with troponin. When it binds with troponin, the tropomyosin rotates and exposes the myosin binding sites on the actin. So the hinged myosin head is able to store energy and connect to actin and pull the actin into the center of the sarcomere. Remember, sarcomere is from Z line to Z line.
There's a student trying to get in.
Okay, thanks. Okay, so if you, oh, there's more. Okay, they missed all the questions answered. We'll do another session. Okay, so everybody's in now. Thanks for telling me, because if I don't hear the little boing, I don't know they're there. Okay, not there. How about here? So we'll keep going. So here's relaxed muscle, the sarcomere. Notice how the I bands are very wide. The A bands include myosin and actin. So when these myosin heads attach to the actin and ratchet or pivot, and they pull the actin into the center of the sarcomere, and that shortens the sarcomere, and we have contraction. Okay, let's look at We talked about that. That's pretty straightforward. Okay, we talked about this contraction process. Okay, so remember, actin is is a connection of globular proteins to make a long filament, and it's has binding sites on the actin for the energized myosin head after ATP has been around and splits to energize that myosin head. When calcium rushes into the all around the myofibrils, the calcium binds to troponin, which causes the tropomyosin to rotate and expose the uh, myosin binding sites on the actin. So, with calcium present and with the myosin heads energized, those myosin heads are able to bind to the myosin binding sites on the actin and they pivot, they're energized, they release ATP, and then they pick up and they detach, and then they pick up ATP, which immediately breaks down to ADP and uh, phosphorus, and the head is energized. So long as there's calcium present and there's ATP present, you can have this incredible event where the myosin head is energized, attaches to the actin filament, pivots, and pulls the actin toward the center of the sarcomere, and then it detaches and uh, gets rid of the ADP and is energized again. And the process happens over and over again, depending upon the frequency of stimulation of the muscle. So that's contraction. So that's what I wanted to cover. So let's go here and let's go back to here. Let's stop recording.